Welcome to my channel XY Biology Academy. Now let's understand each component one by one in its ray diagram. The first component is the laser excitation source. Unlike the fluorescence microscope, which commonly uses a mercury arc lamp, the confocal microscope uses a laser. A mercury lamp produces light of multiple wavelengths that spreads in different directions, whereas a laser produces a highly intense, monochromatic, coherent, and perfectly focused beam of light. Because the beam is extremely narrow, it can illuminate only a tiny spot of the specimen at a time, making very precise imaging possible. Immediately after the laser comes the light source pinhole aperture. This is an additional component that is not present in a conventional fluorescence microscope. It removes stray light and allows only the central, well-focused beam to continue. The light then passes through the excitation filter, which selects the required excitation wavelength, and reaches the dichroic mirror. Just like in fluorescence microscopy, this mirror reflects the excitation light toward the specimen while allowing the emitted fluorescence to pass through in the opposite direction. The next additional component is the XY scanning mirrors. These mirrors are unique to confocal microscopy. Instead of illuminating the whole specimen at once, they rapidly move the laser beam across the specimen in the X and Y directions, scanning one tiny point after another. The objective lens focuses the laser onto each point of specimen, where the fluorescent molecules become excited and emit fluorescence. The emitted fluorescence now travels back through the same objective lens and reaches the dichroic mirror again. Since fluorescent light has a longer wavelength than the excitation light, it passes through the mirror instead of being reflected. It then passes through the emission filter, which removes any remaining excitation light. Now we reach the most important component of this microscope, the detector pinhole aperture. This small pinhole is not present in a conventional fluorescence microscope. Only the fluorescence coming exactly from the focal plane can pass through this tiny opening. Any fluorescence coming from above or below the focal plane is out of focus and is blocked by the pinhole. This is also the reason why this microscope is called a confocal microscope. The illumination point on the specimen and the detector pinhole are placed at the same focal plane, or in other words, they are confocal. Because only light from this common focal point reaches the detector, out-of-focus light is rejected, producing a much sharper image. Finally, the focused fluorescence reaches the photomultiplier detector, or PMT. The PMT is an extremely sensitive detector that converts the light into electrical signals and amplifies even very weak fluorescence. Since the laser scans only one tiny point at a time, the microscope does not capture the whole image in a single exposure like a fluorescence microscope. Instead, the computer records the fluorescence intensity from every scanned point. As the scanning mirrors move the laser across the entire specimen, millions of these individual signals are collected and digitally combined by the computer to reconstruct one complete, high-resolution image. Thanks for watching. Like this video and subscribe to my channel.